Ready? 3, 2, 1, go.
Hi everyone, welcome to today's biology practical session. So today's experiment is very important one as it is there for your exam and it is a compulsory question. It's question number 2 which carries 5 marks, and here you are going to prepare a temporary slide and you are going to study the stages of mitosis, and for this preparation of slide carries 3 marks. And you are going to draw a neat labeled diagram of any one of these stages, which you identify. So the neat labeled diagram of any one of the stage of mitosis carries 2 marks. So altogether the experiment carries 5 marks in your exam and it is a compulsory question for all. Okay. So today let us begin with the experiment. The name of the experiment is mitosis in onion root tip. So mitosis, this is a very common word to all of us. So mitosis is a type of cell division which is occurring in all kind of living organism irrespective of its reproductive method. It could be asexually reproducing or sexually reproducing. We could see mitotic cell division in all kind of organisms. Mitosis mainly help in growth and repair in all organisms, and it also aid in reproduction in asexually reproducing organisms. So, as you all know, mitosis is a type of cell division which occurs in living organisms. So, mitosis by definition, it is equational cell division. Equational cell division means the daughter cells will have the same number of chromosomes as that of the parent cell. Suppose the parent cell is diploid, the daughter cells will also be diploid. The parent cell is haploid, the daughter cells are also haploid. So we call this as equational cell division. So the chromosome number is same as that in the daughter cells with that of the parent cell. So mitosis is defined as equational cell division. Now where do this mitosis take place? Mitosis mainly takes place in both somatic cells and reproductive cells. Mitosis is more common in somatic cells and it will be helping in growth and repair during the life of the organism. So mitosis, any kind of cell division, no matter, it is divided into 2 stages. One is karyokinesis, the other one is cytokinesis. Karyokinesis, division of nucleus. Cytokinesis, division of cytoplasm. So first karyokinesis takes place, that is followed by cytokinesis. Now let me take karyokinesis. Already I have defined it. It is division of nucleus. Now division of nucleus in case of mitosis, it involves 4 stages: prophase, metaphase, anaphase and telophase. The 4 phases of mitotic karyokinesis are in order. The first initial stage is prophase, followed by metaphase, then it is anaphase and the last one is telophase. By telophase, the division of nucleus gets completed and by the end we could see 2 daughter nuclei within one cell. Means at the end of telophase, we could see 2 nuclei within one cell. So that karyokinesis is done, there will be formation of 2 nuclei. So I hope all of you have understood the introduction to mitosis, what I have told. So you know what is mitosis, in which type of cells we could see mitosis, what is the significance of mitosis and what is the stages of mitosis. Now, what is the aim of today's experiment? Today's experiment, we have to prepare a temporary slide by using onion root tip. You all know that the root apex and the shoot apex in the plants contain meristematic cells. Meristematic cells are actively dividing cells that will help in growth. So the root tip, what we are using as a material to study mitosis, will be having actively dividing cells called meristematic cells. So why are we selecting root tip? This is the reason. Root tip contain meristematic cell which is actively dividing, so that I could see the stages of mitosis when I am selecting the root tip of onion. So that's why we are going to use onion root tip for the experiment, and the purpose is to observe the stages of mitosis, particularly karyokinesis, prophase, metaphase, anaphase and telophase. So for the experiment, to prepare the slide, it's very important that we have to prepare the material. So I am going to show you the steps involved in preparation of the material. The material is onion root tip, in such a way that we could see the stages of mitosis clearly and we could differentiate one stage from the other by knowing its characteristics. Now to prepare the material, very important thing, the cells should be visible to us. How to make the cells visible? The cells could be made visible by a technique called staining. Staining, nothing but we are adding color to the material. So here we are using a stain, that is a basic stain, acetocarmine. Using this basic stain, we could color the chromosomes because chromosome contain DNA. DNA is acidic in nature. So once I use a basic stain, the basic stain will have affinity to bind with acidic component, that is the DNA, so that the DNA gets stained. If I am staining DNA, it's understood I am staining the chromosomes. Once the chromosomes are stained, it is visible to us. By seeing the structure and arrangement of the chromosomes within the cell, we can clearly differentiate the stage of mitosis, because every stage of mitosis is different from one another by their characteristic feature. That means arrangement of the chromosomes. Now I am going to explain you the difference in the structure of a cell at each stage of mitosis. Once it is clear to you, it will be very easy for you to differentiate the stages of mitosis. To start on with, the first stage of mitotic karyokinesis is prophase. Now in prophase, what are all the significant features that we have to observe? First thing, here the nucleus gets almost centrally located, and you could see a dotted line. The dotted line indicates disappearing nuclear membrane. So the nuclear membrane starts to disappear in prophase. Then you could see a dotted circle within the nuclear membrane, that is nucleolus. Nucleolus also starts to disappear. So the second feature what you have to observe is disappearing nucleolus. Then once we take the chromatin, chromatin will be thin, long, coiled thread. But here the chromatin gets condensed, it becomes short and thick, and it is now visible as chromosomes. So what we call this as condensed chromosomes, and it is duplicated now. Duplicated means 2n, diploid has become 4n, because it has to distribute 2n to one daughter cell and 2n to the other daughter cell. So finally at the end of this division, as I told you before, there will be two nuclei. Both the nuclei should contain the same set of chromosomes as that of the parent. Now to distribute the chromosomes, the parent cell has already duplicated its chromosomes. So we label this as duplicated chromosomes, and this chromosomes is now thick and short so that they appear dark compared to other cells. Now in prophase stage, you have to notice for a large nucleus with disappearing nuclear membrane, disappearing nucleolus and the duplicated chromosomes. So these are the characteristic feature of prophase stage. And in exam, after preparing the slide, if you identify the prophase stage, you have to draw a neat labeled diagram of prophase stage which carries 2 marks. And the relevant labelings will only fetch you the marks. It's not that you will write cell wall, cell membrane, nucleus. That are not the relevant labelings for the particular stage. The examiner expects you to write the relevant labelings. Relevant labelings for the prophase stage is disappearing nucleolus, disappearing nuclear membrane, duplicated or condensed chromosomes. The next stage, it's very important stage. The significance of metaphase stage is, see, in any tests which is done to detect or study the chromosomes, in particular a genetic disorder, or it may be some fetal diagnosis, or you have to study the structure, morphology of a chromosome, the best stage to study about the chromosomes is metaphase. Very important stage, because in this stage we could see that all the chromosomes will align in the center of the cell. They come and they arrange themselves on the center of the cell, which is called equator. So this arrangement of all the chromosomes in the center of the cell is called as equatorial plate. It could also be said metaphase plate. Now once you are seeing the cell in the microscope, to identify metaphase, you have to notice this arrangement, that all the chromosomes are arranged in the center of the cell, that is nothing but equatorial plate or metaphase plate. So now we can observe here the centromeres are aligned in the center, and to the centromere there is attached the proteinaceous fibers which are called spindle fibers. So these spindle fibers will hold the centromeres in proper position. So the centromeres are in the central line, equatorial plate. That is because of the spindle fibers which are connecting them. So here you could see discontinuous fiber. Discontinuous fiber means the fiber which is connected to the centromere. Continuous fiber. Continuous fiber means the fibers which are passing from one end to the other end without connecting to the centromere. So if you notice this line, it is nowhere connected to the centromere. So it is called continuous fiber. This is connected to the centromere. It is called discontinuous fiber. So we could see both discontinuous and continuous spindle fibers in the metaphase stage. So once you observe a metaphase stage in the slide, in your slide, the clue for you to say the particular stage is arrangement of all the chromosomes in the center of the cell. So this is a clue to identify metaphase stage. And if you draw this diagram with the relevant labelings, you will be scoring 2 marks. Okay, next we'll study about anaphase stage. In anaphase stage, the significant feature is that there is separation of chromatids and the chromatids are pulled to the opposite poles. So in metaphase we saw that all the chromosomes were arranged in the center of the cell. Please notice this. I was calling it as chromosome in the metaphase stage. Now I am calling it as chromatid because the chromosome is a complete structure having 4 chromatids. But now it is having only 2 chromatids. Now we cannot call it as a chromosome. Now onwards we will call it as a chromatid. So the chromosomes which was in the center, now it has splitted. It has splitted at the centromere so that the 2 chromatids on the either side are separated and they are pulled to the opposite poles. So this is what is called splitting of chromosome. Now during anaphase stage, the chromosome splits at its centromere and the chromatids will be pulled to the opposite poles. So the significant feature what you have to notice is that separation of chromatids and the chromatids have moved to the opposite poles. Now once you see through the microscope, you will be observing elongated cell which is having a dark regions in the opposite poles. The dark regions in the opposite poles are nothing but the separated chromatids in the opposite poles. If you observe any such type of cell, then you will identify it as anaphase. And the relevant labelings for this stage is centromere, that is separated. Now we call it as daughter chromosome or daughter chromatids, and then spindle fibers. Now spindle fibers are very important because the shortening of spindle fibers is the only cause that will pull the chromatids to the opposite poles. So these are the relevant labelings for the stage anaphase. Now if you get this stage in the exam, in your slide, you have to draw this diagram with this relevant labelings so that you will get 2 marks. Is now to the last stage of mitosis, that's telophase. Telo means end, the last stage. So this stage is very easy to identify when you are focusing through the microscope. Now in telophase stage, what we observed in anaphase, the chromatids got separated and they moved to the opposite poles. Now here the daughter chromatids which are at the opposite poles, they will undergo decondensation and now they are organized into chromatin. And you will notice the reappearing nuclear membrane. Telophase is nothing but opposite to prophase. Prophase we observe disappearing nuclear membrane. Telophase we will observe reappearing nuclear membrane. And very important thing, there is a formation of cell plate. So this cell plate formation is indication that telophase is going to be followed by cytokinesis, that is cytoplasmic division. Now the daughter nuclei has separated and now within a cell there is a midline formed which is called cell plate, and the cell plate is indication that the cell is going to divide further. That is what is called cytokinesis. So once you observe a telophase cell, the clue is that you will notice the midline which is called cell plate that will separate two darkly stained regions, that is nothing but daughter nuclei. Now we are not calling it as daughter chromosome, it is called daughter nuclei. So by the end of telophase, there will be two nuclei separated by a cell plate within a single cell. So the relevant labelings for this stage are nuclear membrane reappearing, spindle fibers are disappearing, formation of cell plate, very important, and we call chromatin because now they are no longer chromosomes, now they are organized to chromatin. So if you find this stage in your slide, you have to draw diagram of this stage with the relevant labelings so that you will get 2 marks. So in your exam, you have to draw diagram of any one stage that you will show to the examiner. It could be prophase, metaphase, anaphase or telophase. So again I repeat, preparation of the slide carries 3 marks and neat label diagram of any one stage carries 2 marks. Okay, now we will start with the experiment. Before starting the procedure, I'll tell you what are the requirements for the experiment. So as you are seeing here, the onion bulb. So we will take a medium sized onion bulb and we will cut all its old roots, the dried roots what we find at the base, we will cut it. Then you're taking a water in a container. Make sure that the root of the onion bulb should touch the water. If the water goes down, we have to add the water and always ensure that the water is touching the root of the onion. So now you are able to see the root tips which is grown. The onion bulb is kept in water for about 5 days and after 5 days we are able to see the onion root tips. So this is very important. And then we require fixative. Fixative is usually prepared by taking 1 part of glacial acetic acid, 3 parts of ethyl alcohol. So in the ratio 1:3. Now we have prepared it. The purpose of using fixative is that we have to arrest the cell in the stage in which it is. Now for example, I want to study the metaphase stage. I want to study the anaphase stage. So randomly I don't know the cells are in metaphase or anaphase, but we have to arrest them in metaphase and anaphase. So early morning 6 to 8 is a right time to cut the onion root tips and then we have to immediately put it in the fixative. The purpose of fixing the material is that if I leave the root tip as such, it is in metaphase stage while I am cutting it. If I leave it as such, it may progress to anaphase and then telophase. Now by the end, I will be seeing all the cells which are divided are at the telophase stage. It may not permit me to study about metaphase stage or anaphase stage. So the intention of adding the root tip in fixative is to arrest the cell at a particular stage in which it is. So if I add fixative, I am going to arrest the cell in its stage. It is metaphase or anaphase so that I could see all the stages and I could study all the stages. The next requirement is preservative. We can't retain the root tip in the fixative for long. It is only for 24 hours. After 24 hours, we are going to take the root tips from the fixative and then we are adding it in the preservative. Here the preservative is 70% alcohol. Now once we store the root tips in the preservative, we can use it for many days. Then for the further experiment, that is, we require one normal HCl. One normal HCl is required while we do the experiment. That means while preparing the temporary slide. We are going to use one normal HCl and we are going to heat the material with one normal HCl. This is done to solubilize the gel which is present between the cells. See, intercellular spaces will be filled with some gel-like material. We have to dissolve that gel-like material by heating with acid. Once we do that, the gel gets dissolved and the cells will get separated. If the cells are separated, it is very easy for us to study the cells. If all the cells are clumped together, they are in group, we can't study a particular cell at its particular stage. So the purpose of using one normal HCl is that we are heating the material with one normal HCl to dissolve the gel present between the cells, and that allow the cells to get spread and also to soften the material. Next, we need a stain. Very important. Acetocarmine. I already told it's a basic stain. If we add this stain, it readily binds with the DNA acidic component. That means we are staining the chromosomes so that the chromosomes become visible. Then we need a microscopic slide, cover slip, brush, needle, watch glass and blade. Okay, now let us study the procedure stepwise to prepare a slide, temporary slide of onion root tip to study the stages of mitosis. So to start with, first step, you are taking one normal HCl, 4 to 5 drops of one normal HCl in the watch glass. So now, after taking the HCl in watch glass, we have to take a root tip which is preserved. Using a brush, into the watch glass containing one normal HCl. Now I have taken the root tips into the watch glass containing one normal HCl. Next step, we are going to heat the material. So now you hold on with the forceps and we are going to heat the root tips. So heating is very important step. You have to heat it thoroughly. Once heating is done perfectly, the cells get separated and we can study the stages clearly. And don't heat constantly. Heat for some time, 5 counts. Take it back. If you constantly heat, the glass may break. So that don't heat constantly. Heat for some time, allow it to cool and then again proceed with heating. Take it. So this you can count 5 counts 5 times. Do not heat it constantly. So now the material is heated with one normal HCl. Now using the blotting paper, drain out the HCl in the watch glass. Just drain out the HCl completely. Now what is left out is only the root tip. Now to this I am going to add acetocaramine stain on the material and leave it as it is for about 20 minutes. So this is staining, it takes some time. So after 20 minutes we have to take the root tip on the slide, place the cover slip and gently press it with the thumb. Okay, now after 20 minutes the root tips are stained. So I am going to take it on a clean glass slide. Using a brush, I have taken the root tip on a glass slide. So the tip will be darkly stained. So you can differentiate the tip at this side towards the left side. It's darkly stained. I am going to cut it half because as I take a long root tip, more cells will be seen. So these cells are not necessary. The cells which is towards the tip is required for our experiment. So that I am going to cut it just half using a blade. This is just to reduce the cells within the slide. Now just take a stain around the material. Now we are going to place the cover slip. So to place the cover slip, make sure you are not leaving any air bubble. Just take the help of a dissection needle, place it oblique to the needle and gently cover the material. Now I have placed the cover slip. So you have to blot the excess stain by just folding the blotting paper on the slide, and so that the excess stain will be drained out like this. And the material is also squashed. So if you heat it thoroughly, just if you press, a gentle press will squash the material and the cells get spread. Now the slide is ready to focus. So we have to focus it under 10x first. Then we have to focus it under 40x to see any one of these stage. So now the slide which we have prepared is ready to focus. So we have to focus it under 10x first. So now I could see the cells, the root tip cells which I have spread and they are stained. So I can see darkly stained nucleus within it, but stage could not be seen in 10x. So we have to increase the objective of the lens so that now I am going to focus it under 40x. No need to move the slide. The slide is fixed in its location where I could see the cells properly. Only thing, I am going to raise the fine adjustment and then I am going to change the objective to 40x and focus it now. Okay, now under 40x I could see the cells, dividing cells, and more common prophase cells are seen. So the metaphase stage will be having the chromosomes in the central line. Anaphase cells will be showing the chromatids in the opposite poles. telophase cells will be showing the central line which is called cell plate. So for each stage, the clue which I have mentioned while explaining the stages, so you have to keep that clue for identifying the stages. So you could show any one stage and draw the diagram of the particular stage. So the experiment gets completed while you show the stage to the examiner. So slide preparation carries 3 marks and the stage diagram carries 2 marks.